Okay, this is the 28th. I have a student. She's agreed to be video recorded, and we're going to be going over old tests today. So, or lecture notes or anything. So, questions. Let's go.
I don't really get how students are like asked.
So, which one?
Like on the homework, I don't know how mine is.
Okay.
But like the last column on the first page.
Where you're doing the pico green?
Yeah.
Okay, I have my old one, so I'll grab it. Okay, so remember with the first die, it's going to be, you're only going to see your 5' end. But with the pico green or ethidium bromide or something, let's see, do you want to write on that one, or here, I can write on this one. So let's this one. I'll do this one. So with pico green, or for the heavy phosphorus, you're only going to have one here. So if it cuts here, then you're not going to see anything. So with the P32, you're going to see mainly everything that's cut from the right. And so that's what you're seeing in this line here. But when you do the ethidium bromide, you're going to see everything because it intercalates, which means it gets in the, do you remember if it's a major or minor groove? It's a major groove. But he probably won't ask that. But it'll get into, just like slide into the major groove, and so you're going to see one at every cut point, or at every bond here. So to begin with, you're going to see at least the same as, is this one the same? This one isn't the same. Oh, we can do the homework first and then move on. So you're going to see at least the same as the P32 die. We know that because that's going to be everything cut from the right. So we have at least this, because this is going to be our ethidium bromide. So what else are you going to see, do you think? So remember, this is our normal protein here, or our normal protein, and this is from the shift of the protein. Whenever you cut it, it's going to make the DNA heavier. This is with nothing bound. So, any ideas of what you're going to see? Okay, so for the one with it bound, if you cut it, so the bottom two here is just going to be this first cut, and that's going to be your first one, and then your second cut, and it's not going to be able to cut for all of the red, right, because it's going to be blocked by the protein, and that very next cut is going to be this top one. Okay, so, but what's different is that we can see everything cut from this way. So if we cut here, it's going to be, so with the ethidium bromide, if we cut here, we're going to have a one length and then this whole length. But that's going to be the same as this band, isn't it? Because you're going to have, if you cut here, you cut off just one off the end, and then you have this whole length, and you cut this one, you're going to have one in this whole length. So that's going to be the same as this one. Same with the second cut. If you're cutting it from this way, then you're going to have the two and then the whole shifted one. So you're going to have the two and then the whole shifted one. So that's where those two are from. But what we're going to see that's different is going to be another cut here, 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 and here. Because we're going to have unshifted 3, right? Because you're going to have your 3, unshifted 4, unshifted 5, unshifted 6, and so on until you get back to here, and then you're going to have your shift. So does that make sense?
They're going to overlap. There's going to be something in everything.
Yeah. So we have our unshifted 1, which we have, our unshifted 2, which we have. I'm just going to draw a dotted line so I don't, like, do them all over the place. It's like my underlining. Whenever I try to underline things, it always like I'll end up going and I'll be like, like us.
You don't even want to see that.
So we're gonna see an unshifted 3, so that'll be right here, and then an unshifted 4, unshifted 5, unshifted 6, unshifted 7, unshifted 8, unshifted 9, unshifted 10. We'll also see unshifted 11, but there's already one there, unshifted 12, and unshifted 13. Up to here, because that's just cutting one cut, cut. That makes sense.
Yeah.
But whenever you have a gel, the doubles don't show up as anything different. So on this problem, it's just going to be everything. Everything's going to have a band. That's good?
That's good.
Okay, so we can move to a more tricky one. And I'm going to use a different one to make sure that I do it right. Because it's just a thought experiment, that's all it is. Okay, so this is where the protein binds here. Just do you get how to do where that binds? You just count the bottom and subtract. So that's where it binds. And we can check 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Okay, good. So, what is the first thing we're going to do?
Everything that was in the third column.
Yeah.
Good job.
Yeah. So, here, you can do it because I can't do upside down. So the same thing as the third column. Okay, so next we're going to count from the right and do the unshifted. So we're going to have unshifted 1, which we already have. 1, 2, 3, 4, 5, 6. So does that go up to 6? 1, 2, 3, 4, 5, 6. Okay, now we can also start from the other side. Does that make sense? Or actually, let me see. So, what else could we get? So, cut here, get that one. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So what else you're going to see on this one, it's kind of harder to see. But, so this top shift is 1, 2, 3, 4, 5, 6, 7 is going to be the 1, 2, 3, 4, 5, 6, and then the 7 here, because you're going to see that one. So with the other die, you're going to see if you cut it here, that's going to be 1, 2. That's going to be shifted, all of them except for 1. So that's going to be your top one. So that's going to be, and then the shifted 2 is going to be the bottom, the next one. Does that make sense? So if we're cutting, let's see, pretend this is a protein. Remember the trick with this is just to not get like all crazy about it, because it's just another thought experiment like the other one. So if the label is attached here, right? And so if we cut this one off, it's going to be the same as cutting this one off. So that's going to be the top one, okay? So one more is going to be the next one, okay? Do one more, this is the next one. One more 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 is the next one. I skipped a shift. But we're going to have one more there, and because I skipped a shift, it's going to be one more. Let's see, so where else would we see something that's not? So we're going to have more unshifted, or more shifted up here. So we're going to see one at 6, 7, 1, 2, 3, 4, 5, 6, 7, at 14. 2, 3, 4, 5, 6, 7. So it should be right here. And so we're missing 2 lines. So where could those 2 lines be? Let's see, did I draw this right? Yeah. So if I cut there, cut there, cut there, cut there. So be all those. Oh, also, I forgot this trick. So, if we cut here, that's going to be the 1, 2, 3, 4, 5, 6, and then the shift, right? So that's going to be same as, on this one you have to kind of calculate the shift. So the way to do that, you know your molecular weight here is going to be 20, right? Because of your marker, so 20. And this is going to be 24. So the way to do that, then you know the shift is going to be 4 units, kind of. The Svedberg units, remember? And so we can kind of roughly add them together. So you're going to have your 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, plus the 4 from the shift. So that's going to be 16. And 16. And then you can subtract the 16 from the 24, or from the 20. And that's how many extra bands you're going to get. I think there's a formula. Was there that formula in the book?
Yeah, possibly.
I had a better way to explain this last week. One sec. We cut there. So that's going to be 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. We're going to have 1 extra there. 1, 2, 3, 4, 5, 6. Let me see. So this one is going to be 1, 2, 3, 4, 5, 6. 6. So it should be at 14. So I think we did it right with that subtraction. When you have to incorporate the weight of the shift in it. Okay, so let's try that one. She got it right?
Yeah.
Okay, so, and there's probably another one in that one?
The year before her.
Okay. I think mine is the same as one of the homeworks, so that's not too helpful. Or it might not even have. Oh, actually, we have the same one. 1, 2, 3, 4. Okay, so I'll erase this because I have the same one as her, and so you can keep that one. Okay, so our first number 1 thing. If you get some of the bars, you get extra points, or you get more points than if you just get none of them. So first thing, what do you do? Oh, so this is no protein. This is protein plus the phosphorus. So the first thing, remember, is to do the same as the other one. Okay, so there you go. Go for it. I think this one's actually easier than the homework. So then which ones are going to be unshifted that you'll see otherwise?
From this end?
Yeah.
So 1, 2, 3, 4.
Yep, and that's where the protein is. I got it wrong. So yes, you're going to see 4 unshifted, right?
So they go in here.
Yep, just right there. And it'll just line up with those 2. There you go. And see, so will we see anything else? That's the question. So let's say we cut this one, cover up cut off the dye there. It'll be the same as cutting off this one, right?
Yeah.
It'll be the same like another way. So if you cut off the second one from the left, yeah, it'll be the same as cutting off this one. So we'll see that one too. So let's see what else.
Let's see if we put it here. Would we see this side?
No, because remember, I don't think you can cut that close. It cuts on this side, but not this side. Yeah, it doesn't cut at that point right there. It does cut off this side. Um, but even if we did cut there, so you'd have your 1, 2, 3, 4 plus your shift. So 1, 2, 3, 4, 5, 6, 7. So it's 7 plus your shift, which is going to be pretty big. So say your shift is, what is it? 1, 2, 3, 4. 2, 3, 4. It's going to be 8. So this is 8 here. This is 24. So your shift is going to be like 16. Plus another 7 is going to be at the top somewhere. So it's not going to be in between. I can't do that. So it's going to be at the top somewhere. We don't have to worry about it. But other than that, I don't think we have anything else. Because if we cut 1, 2, so the shift, if we cut that one, it's going to be the top, that one's going to be the top, that one's going to be the top, that one is going to be the top. Those are our 4. Oh, and then the last one, there are 5 at the top. And that's all we can really cut. If we cut that one, it's the same as that one. If we cut this one, it's the same as right here. So, is that what you got?
Yeah.
Yay! So I thought this one was easier because in this homework, it's the longer it is, kind of the more tricky it gets. Let's see, 1, 2, 3, 4, 5, 6. So that'll be the same. If we cut here, it'll be the same as 1, 2, 3, 4, 5, 6. We'll have 2 extra for sure. Well, I'll look at this one again. Did you get it on the homework? Or he hasn't given it back yet?
No, he just
Okay.
got it first.
Because I had a good way. I had a really good way of like a math equation that worked really, really well, and it made a lot of sense. But I'll think about it, and if I remember it, I'll email you. Man, I can't believe I forgot it. I should have written it down. Oh, well. Yeah, I'll be sure to email you that. Okay, so do you have the other one from the, or is it the same?
I don't know if there is.
Is there? Two, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. Did they have one on there?
No.
Okay. So do you just want to go through the old test and hash out the problems? Let's see if I can help.
This is the same. This one which is the.
Okay. And these are just the...
What's the operator and the oppressor?
Operator is... those are for eukaryotes. He hasn't talked about it in class. It might be in the reading he gave you last time, or the one that he will give you. But we haven't done a lot with eukaryotes, and that's what it is for eukaryotes. And is this upstream or downstream of start? Why? Because it says downstream? Do you remember the numbering? How you number?
These ones are positive and 4 start is negative.
Yeah. So it's perfect. So like on these are all negative, so they're going to be upstream of start. Yeah, negative again. This is the same problem, so they're still going to be upstream. So yeah. So just look for the sign on those. And the final lane, what does he say? What would you expect to see if you need a... Okay, and we got that one pretty much hashed out. I think that for this test, Gabi's going to do his own review also. Because, yeah, on our test, the average was pretty low, so I think he's worried about it. So he'll give you extra help. Oh, this one looks fun. What is this? Suppose we isolate 3 proteins that we said. We have 100. Okay. Oh, that's exciting. Goes to protein 1. Okay. Oh, this is cool. Okay. So there should be, let's see, so on the first one, are there 20 here? That's right, there are 20. Okay, so the next one, you only have one protein. So it's just one protein at a time, it looks like. And what? Only one of the proteins. Okay, so you're going to have one protein for each strand, it looks like. So we can do our subtraction trick. Remember, so you count the bottom ones. 1, 2, 3, 4, 5, 6, 7, 8. 9, 10, 11, 12, 13, 14, 15. So it's going to be a 5 mer. So, and then it'll also be 8 up. So it's going to be, you're going to see 1, 2, 3, 4, 5, 6, 7, 8. And that's where it's going to bind to 5. 1, 2, 3, 4. Oh, wait, did I count one? It's 15, right? 1, 2, 3, 4, 5, 6, 7, 8. And 1, 2, 3, 4, 5. Oh, yeah, you don't count this first one. I keep on forgetting. Okay, so then the next one.
Wait, which ones do you count?
So it's gonna be, you count the total, remember, and I think it's 15. 1, 2, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. And this one was 20. So you know it covers 5.
So
you know it's 5 long.
So these ones are 8.
So let's see, if you cut here, that's going to be your 1, your 2, your 3, your 4, your 5, your 6, your 7, your 8. And then it's going to be obscuring this cut side. So it's going to be obscuring 5 cut sides. 1, 2, 3, 4, 5. Okay. And just try to visualize what's actually happening in the gel, or what's actually happening. So you're gonna see it when this side is open, like so when this is active. Does that make sense, or is just more? Okay. But it's essentially measuring the cut sides. That's what you're looking at. Oh, this one is kind of neat. So this one is going to be, let's see, so -4 is going to be 6. So this is going to be 16, right? So you know it's going to be 4. 4 long, you can also see this is 4, and the second line there is 4. So it's going to be, oh wait, yeah, I cannot count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. It's going to be 2, not 4. So you see there's nothing else down here, so you're going to be hiding the first site and the second site, and then we're up to 18, so that'd be the rest of it. And this one's kind of cool. We never had this one in class, so it's neat. So how many are there? Let's see. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So 5 are going to be covered total. We know that because there's 15 and then there's 20. So our first site is going to be 1, 2, 3, and then we're going to, how many are we not going to see? So there's going to be 1 here, which is good. Let's see, and this is where you would look at the shift. You just subtract this top line. Its molecular weight is, what, 23, I think? The top line, what is the weight of it? Can you see? I can't see like sideways. Is it this line that's the top of this one?
Yeah.
Okay, so that's going to be 22. So we know the shift, and this is 20, so the shift total is going to be 2, right? Because it's this top weight minus this bottom weight. So you can shift this one down 2 also. So what is this weight here? So this is, can I write on it? And we can erase it later. So this is going to be 22. This is going to be 20. So the shift is going to be 2. And what is this weight here? Can you see? I just can't do it sideways. Here we can use, if this is kind of straight, it's not really all that straight. Here. Okay, so that's gonna be the 1, 2, 3, 4th line, right? 4, so that's gonna be 8. So it's gonna 9, right? The weight here.
Yeah.
So that's a 9, and then minus 2 from the shift is gonna be a 7. So then that's gonna be your 7th one. I think, yeah, it's gonna be your 7th. 1, 2, 3, 4, 5, 6, 7. That's where it's gonna end. So that's good. That's where it ends here. That's what we're gonna see next. And you can do the same thing. So where would this one be? Yeah. So or you could just do 7 plus 1, 2, 3, 4 cuts. So 1, 2, 3, 4. Oh, that's weird. Why is that wrong? Oh, okay. I think that's right. 11. Okay, what is? So let's just say this is 7 and 11, and what is this weight here? I can't see it. Here, use our fancy paper tape. One of the sides isn't straight, so be careful. So 13. So that's going to be at 10. Wait, this one's 13. This is 13, you said?
Yeah.
So 13, so it's going to be at 11 also. Do we count wrong? So 11, 12, 13, 14, 15, 16, 18, 11, 12, 13, 14, 15, 16, 17, 18. So that's going to be our 2. So there's definitely 2 proteins. This one's weird. I'm pretty sure, so we know this is going to be a 4 right here, and it's going to be shifted. So 1, 2, 3, 4. I'm pretty sure this is what it's supposed to be. I think this X is where Gabi was marking where it was. But it looks like she scribbled it too. And then from the top we have 1, 2, 3, 4, 5, 6, 7, 8. So that'd be 1, 2, 3, 4, 5, 6, 7, 8. So maybe you see it here. So all of them, it's gonna be 1, 2, 3, 4, 7. I think it's supposed to be shifted down right here. Because from the top you see we have 8 bands. So that's going to be saying, oh, we cut here and we see all this. And we cut here is going to be the second band from the top. So I think you might want to ask Gabi on this one because I'm actually not sure. But it might be here. I think this is what it is here. And he might ask something like this on the test because this one's pretty clever actually.
So you know we didn't talk about it in class. They could bind it to different places.
Oh yeah. You could have protein C. This is fairly common actually to have proteins that kind of jump. I mean, think of like RNA polymerase. If we stuck that on, that's what it would be, and we froze it. That's what it would be like. It'd bind at one spot, and then it'd be at another spot also. So
you would expect us to like know that or like figure it out.
Yeah, I mean it's basically the same principle. You look at the shift, so you know the shift is going to be 2 if we counted right, which I think we did. Yeah, the shift is going to be 2. These are going to be unshifted, and this one is going to be shifted. So 7. Let's try to draw it unshifted actually. So without a shift. So 1, 2, 2. Okay. But it's not because I can't draw straight lines. So one thing that helped me on the test is to, like, I got a piece of paper like this and I just drew the unshifted ones, so that'll show you like where they actually are in the 1 through 20. Because all this shift is like, remember the fat man with the monkey? This is the monkey. This is what the shift is doing. It's a monkey on the back. So
Wait, so could it's still gonna cut like in between the two places?
Mhm.
Would they still be attached though because of the protein? Or...
That's a good question. Let me think. Technically, yes. But it depends on what protein you're using. Oh no, I guess it shouldn't matter what protein you're using. Because these are... It's cutting those phosphodiester bonds. I don't know. That is a good question. I think it's supposed to be where it just cuts the protein too, but I don't know what it's cutting in the protein. Because the depending on what the digester is, it could be cutting like the phosphodiester bond, or it could be cutting off part of that ribose chain, like part of the ribose. It could be cutting part of that. Which could technically cut part of the protein because, you know, the protein is just amino acids, so that might work. But it's a good point because then you don't know how big the shift is if it cuts in the middle somewhere.
So like, since it shifted to like one of them maybe here, maybe here or something.
Yeah. That's a good. I'd go ask him on that one. So I've never seen anything like this. But the first two are pretty obvious, right? Just because, and seeing this one, he didn't ask for the other one. No, that one's tricky. Tricky. I wonder what it was actually here though. 1, 2, 3, 4, 5, 6, 7, 8. See, I would have drawn it this one. Because then you'd have 1, 2, 3, 4 here, and then wait, here, and then 1, 2. That's interesting. Yeah, I go see him about it. Sorry. Name the molecule that permits us to visualize our DNA fragments. Do you remember which one? Stop looking.
P32.
Yeah. There you go. And what others could you use? For your intercalating, what other 2 other dyes do you remember?
Is that pico green or is that a different thing?
Yeah, pico green is one of them. Ethidium bromide is, yeah. So those are the only 2 intercalating ones that we know of, or that he told us. So that's a good question on this one. I'll have to think about it. Maybe bring it up at the review session, or if you don't want anyone else to know about it, you could just give us all the respect. Oh well. So let's see. Is there another specific one that you wanted to look at?
No, I haven't really looked too much at this one.
Okay. So draw A and T base pairs. That's just something you have to memorize. Sorry. We could practice drawing it if you want.
I drew so many of them.
Mhm.
Like I have like one color for A and one color for T, and then like a different color to do bond between them.
Yeah.
It's like because I told him I was like reviewing that, and he's like, "That's a good thing to know for the test." So I'm guessing we're gonna have that.
Consider the following things of human DNA. Circle the promoter. It's just the Tata. And remember the consistent. It's good to remember the consensus sequence. Tata. Remember then AT, AAT. That's the consensus sequence for Tata. He should have told. Oh, for human DNA. Okay, so be careful with human DNA versus like bacterial DNA because it's different for prokaryotes. For prokaryotes, I think like the Tata equivalent is the Pribnow box, if I'm remembering correctly. Do you remember? Or is the Shine Delgado? So yeah, that's Tata. See, I don't think he might not have gone over the prokaryote promoters yet. So yeah, it's fine. It's in the textbook. So.
Yeah, I don't have anything.
Okay. Well, I'll pull it up in the textbook real quick. Enhancers. Oh, yeah, so in prokaryotes, it's the Pribnow box. Tata box. So promoter. So transcription factors in eukaryotes are they're called transcription factors. It's what binds to the promoter. In eukaryotes, it's sigma factors, which he hasn't talked about yet either. But, yeah, so Tata box is eukaryotes. Pribnow box is not prokaryote, or it's prokaryotes. This is eukaryotes. I might have said it differently. Yeah, and it's important to know these numbers too. I can go over through them. 35 to 10 is kind of wait. Oh, so at negative 35 to negative 10, you're going to have your sigma 70, which is kind of like a Tata box, and it's Tata AT for prokaryotes. Eukaryotes is almost always Tata. But so this is kind of tricky. And numbered like at minus 10 and further, it's the Pribnow box. Or if it's at minus 10. So just be sure you know, because on my test, this was a question. He asked us like, oh, if this was a prokaryote, what would it have been? So be sure to know those. Also, those numbers.
So do they do the same thing as the Tata box?
Yeah, they're almost identical in function. They both bind transcription factors or sigma factors, and then that recruits the RNA polymerase. Like either 1 or 3, depending on which thing you're in. So, okay. So what else? I like the old tests; they're really fun. Indicate the transcription start site, and that's just because it's 25 downstream Tata is. Oh, is that what he said? Yeah, it is. It's just numbered negative 25. That's a good thing to know. Negative 25 to negative 35. But it's almost always negative 25. So, place the box on what corresponds to the stop codon. Remember, stop codon is AUG, so then you have to translate it into AUG, A to T, and U to A. Stop codon. I'm getting start. So yeah, so this is stop UAA, UAG, UGA. And so you have to look for either a T and then, or actually, yeah, a T, because that's what it's going to be, and then AAG, but it's here, and then AGA, so it's going to be T, it's going to be this one, TAG. Because this is DNA, not RNA. Remember the stop, your 3 stops. This is what 3 stops are in RNA, or mRNA. Okay, then is this the polymer? He hasn't talked about that yet. I haven't looked in the textbook. He might give it to you tomorrow, but that's an important thing to know. But he hasn't talked about it yet, so off the hook.
For now.
Given the recognition sequence of Hindi 3, oh, so this is tricky. So Hindi 3 is a palindrome. Remember, that's forward as it is backwards. And that's AAG. So it's AAG going this way, and then remember this way, it also has to be AAG. So AAG, then TTC, because that's what it has to bind to there. It starts with AAG, and it cuts between the 2 A's. So you have a cut here and a cut here. Does that make sense?
Yeah.
You see how you got this CTT? Because it has to match the AAG going the other way. Write out the entire recognition sequence. indicate what the results will be. Okay, so this is good. He tells us between the two A's. So would this be 5' or 3' overhangs? You see, this is where that's where it's separated. You say that like a question.
It is.
Yeah, so what helped me the most, it is a 5', you're right the first time, so that's good. It always helped me to write what each strand was. So this is going to be 5', right? And this is going to be 5' here, and then that's a 5', and then 3'. And going this way, it's going to be 5' and then 3'. So I just looked at whatever was at the furthest end, that's what it's going to be. So this is going to be 5', and this is going to be 5'. There are ones that are 3', so, and on my test, I think the first question was, is this 5' or 3' overhead? So see, this is my test, is the first one. I should have just labeled it like I just showed you. But see, going this way, this is a 5' here, and going this way, it's a 5' here. So this would be one sequence, and this would be the other one. So it's a 5' overhang. I think it's also Hindi 3 actually. Yeah, Hindi 3. So be sure to write it out. Don't trust yourself. Okay, what's next? Do you get this one, or do you want to go through it? Yeah. Okay, so the first one, uncut. What are you going to see? I know it's on the test, but what do you think you would see? Would you see one at 6,000, or would it be higher than 6,000 or lower than 6,000?
Could it be because it's like,
It would be what?
would it be at 6,000 because it's just 6,000 and it's not that anywhere?
Well, remember, that would make sense, but
Or
that's
is
not.
It's the circle, so it like doesn't.
Yeah, it coils and it gets really, really compact. And he wrote super coiled, and technically it should be a little bit lower than 6,000, so it should be, that might be it a little bit lower. He said 6,000. What line is that? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 6. Oh, so that's right. So this is at the 5,000 line here, but it's 6,000 molecular weight. So yeah, just anything lower than 6,000. It's not going to be like 100, but it's going to be like maybe 4,000 or 5,000, just because how it coils and packs in really tight. So it only will show its full length if you cut it once, because then it can't coil correctly.
Like the beads when he like dropped one edge.
Yeah, just like that. I had forgotten he did that. Yeah. And the next one, NLA4, it looks like. So what would you get there? Where would you cut it, and what would you get?
So it's that one.
So what would your fragment be? No looking. I'm just gonna cover this up. So you're just gonna have. It's only gonna be 6,000, right? Because there's only one NLA. Be sure to check this, because this is what got me on my test, is that I just got blind or something, and I didn't see the other one. And so NLA, make sure that these are not, there's only one NLA. So you only have one cut, which is going to be, like you said, the full 6,000. The next one, Bagel 2, it looks like. Is that what it says? BGL 2. So BGL 2, you're going to have one here. No. So what would it be? Cut at 1,500. Nowhere else. How long would your strand be?
6,000.
Yeah, so it's also going to be 6,000. And NLA 4 and Bagel 2, what are you going to get? So you're going to get...
So, like 1,499.
Yeah. So you're going to get one at $1,499, and then at $1,499 minus 6,000, whatever that ends up being. I'm not good at mental math. But okay, so then Nico 1, what are you gonna get?
So there's 2 cuts.
Mm-hmm.
So that minus that.
Mm-hmm. Yeah, so you'll just subtract this one, you'll get 1 length, and then you'll just subtract that from 6,000. I don't know, did he get that right? Nickel 1. Is that right? 1766. Now let's pull up the calculator real quick. Calculator. What were the numbers for the cut sides? What should I subtract?
45, 78, and 28, 12.
8 minus 28, 12. 1766. Yeah, so that's right. And then 1766 minus 6,000. Oops, not 600. 42, 34. Should be, and he got that other one. Yeah, oh, he did get it right. 42, 34. There you go. Let's see. Messed up. Well, okay, so that's good. That one makes sense. Nico 1 and P3. So you're gonna have your Nico, the same Nico band here, right? So this is gonna be your 42, 34, and you're gonna have what next?
This one.
This one. Yeah, and so you just subtract again. Easy math, right? Don't be deceived. Recheck, double check, triple check.
Bring your calculator.
Yeah, for real though. Bring your calculator. And then Nico 1, and also be sure that you don't get confused at 1. Because one thing that a few people in my class did that we were talking about is that they started at 1. So they said for like the Nico cut, it'd be this one, and this one, and then this one. That's wrong. It's this whole one, and then this one. So just make sure you don't confuse yourself. And now that I've mentioned it, you'll be thinking about it and you'll confuse it. But yeah, so did we do this one? Nico 1 and Plea 3. So Plea, oh, we just did that, yeah. So this one, that one, and that one. Nico 1 and BSE 1. So you're gonna, what are you gonna get?
This one. So
Uh-huh.
it's the 1766.
Yeah. And one of those, whatever those numbers, I guess you got them all right. Yeah. So that makes sense to you. I have another one on my test if you want to do it. It might be the same one actually. Do you want to do it? Okay. Oh, it's different. You see, this is me not doing mental math. So uncut, I just put question mark around whatever that is. It'll be tightly coiled, intact. 1 cut for NLA 4, right? Just 1 cut there. Bagel 2, just 1 cut. Yeah, just 1 cut. NLA 4 plus bagel 3. So this is going to be 1,500. See, I cannot do math. And then the other one, so that one, and then that big one. So 5,500 just minus 1,500 is going to be 4,000, right? Not 39.99. So this is important. Just don't think about it. Just subtract. It's easy subtraction. Nico 1, what are you going to get? Yeah. So be careful on, I just kind of did something I thought about, be careful on this. And if you have to do a cut that's like this, that you have to figure out what this length is, and then this length also. So it's not going to be 47.5. I mean, for example, if you do this one, it's not going to be 47.5 minus 2,800 this length. That's going to be wrong. You have to do 47.5 or 40, this one to this one to this one, because it starts over at 1 here. So just be careful. Yeah, so that was right. And then I think you got it. I think you're good on this one. Okay. So next question. That he hasn't gone over. We had the same thing. Gene delivery vehicles. He might do that next class or the class after, but be sure.
It's got the gene delivery packet.
Oh, okay. Did you get the one that's, it's a big book? Let's see. It's this. This, have you gotten this yet? It's just a lot. Be sure you read through this because this is where the Pan Am. I don't know if it's in the textbook. I haven't looked at the textbook yet, but this is like one of his students wrote this, so he really likes it. Yeah, see, WT. But, that's where this is talked about here. What's the next one? The reasons why permanent. Okay, he hasn't gone over that. He hasn't gone over that. He hasn't talked about that either. So you're good on that page. Since you don't really call DNA and plasmid DNA or both DNA, how are they separated during the plasmid prep procedure? And that was last class. Just you have to remember the 7 or 8 steps. He might ask something about the charge for that anionic exchange, but just remember they're anions, so they're negatively charged. But yeah, those steps, you have to know. We didn't even get, whenever I took it, he didn't give us these steps. He didn't have a question like this. We didn't even talk about it. So, okay. And suppose you have 2 plasmids, one can you insert from the one. Okay, so how would you do this one? Do you remember if your cut sites don't match up? How do you overcome that? Yeah, blunt end ligation. And so the 2 enzymes, you're going to have your ligase, which is going to stick it together. And there's the ligase, and I think it's one of the polymerases will get the other stuff. But that's all in the textbook. So just make sure you understand blunt end ligation and just in general, how effective is blunt end ligation versus sticky end. Yeah. So it's maybe 1 in 1,000, something like that. Maybe 1 in 10,000. That's not very good. This you haven't gone over yet, so you're good. It's kind of a fun thing. There's AB and ABO to talk about. Yeah, it's cool. I mean, it's just a Punnett square kind of thing. So the A's are either going to be AA, like A capital A, or A like recessive I. So, or AO as he said. And O is just going to be like I. But he hasn't talked about that, so. Unless it's in the textbook, then you should know it. Yeah. And that's all. So you're going to get an 83 on this test? You can do it. Okay. Okay. Let's see. So that's draw the separate fragments as it would result from the exposure of the following. Yeah. So are those going to be 5' or 3' overhangs?
So this is 5. This is 3. So this is 5.
Yeah. Perfect. Draw the results after falling DNA fragment is exposed to the Klenau fragment. Remember, the Klenau fragment is going to be 3' exonucleases and then 5' polymerases. They're really tricky. So everything at the 3' hangover is going to be cut off until you hit a double strand. So that's what's happening here. So this is your 3' end, right? So 5', 3'. So if it's going this way, it's going to cut out everything. Sorry. It's going to cut out everything until you hit your double strand. And then your 5' polymerases is just going to fill it in. So I think that's a giveaway question. Okay. What is the next one? The enzyme Lu1. I've never heard of that enzyme. Okay. Has a recognition sequence if it is used to complete how? 2 5' overhangs. Okay, so that's just kind of the same thing you did up here. especially if it's the same enzyme, then they're going to be the same kinds of overhangs, because it's just going to be like this. So you're going to have this end, and then on this one it's going to be another 5 prime, because one enzyme only produces one kind of overhang. So like for example on this one, 5 prime overhang, 5 prime overhang, it's not going to switch. So if you're using one enzyme, it's going to be only one kind. One prime, okay.
So that automatically eliminates like half the answers.
Yeah, but it's those few answers.
And then 2 primes.
Okay. The endotoxin removal step in the plasma protocol involves a proprietor. That's the one that you're supposed to look up, and that he'll. Did he? I don't know. I didn't have this question whenever I took it, so sorry. But look that up on the internet for sure, because he was like really stressing it in class. And it's not in the textbook either. So he told me actually that I'm not allowed to tell y'all, or not. I mean, he wouldn't know, but I'm not going to. But he said that it's good for y'all to look it up. But yeah, so be sure to look that up. He asked, I think, how do you get out the RNA, and then how do you get rid of your protein and your endotoxins, right? I think those were his. Or he had 2 questions that were like, oh no, these.
So which reagent to remove the endotoxins?
Mm-hmm.
And he said it would prevent the plasmids from binding to the antigen.
Okay. Good. I don't know. Yeah, that class I was kind of sick, so I wasn't like all the way there. So sorry.
That's okay.
Let's see. So this one is the same as the other one. And that I mean that you haven't gone over it. Besides each item indicate DNA, RNA, or neither. What? Sorry, my neck is getting tired. Molecular weight, no enzyme. Oh, okay. Soon, RNA? I don't know. This is a DNA plasmid. I don't know what he's talking about, if you're supposed to indicate if it's DNA, RNA, or neither. But if you have something circular, it's almost always going to be DNA, your plasmid that you're going to insert, or that your plasmid in your bacterial genome. But do you remember the general differences between DNA and RNA? There are like 2 main ones. Or 3 main ones. Okay, so DNA, the first 2 are going to be structure. So structure-wise, what's different between DNA and RNA? Hint D versus R. Yeah, so one of them is going to be your OH. The next one is going to be in the bases. What's different about RNA bases? Yeah, instead of T. Close. Closest though. Your third one, DNA versus RNA, has to do with processing. What would DNA have, or what would mRNA have that DNA wouldn't have? Hopefully. So remember, whenever you process RNA, what are you going to have on the tail? Yeah, your poly A tail. So DNA could have a poly A section, but it's not very likely. And what's going to be on your 5' for RNA? Yeah, which is going to be, I remember it's M7G. Do you remember what that stands for? It's going to be it's methylated guanosine. And methylation, he hasn't talked about this yet, but methylation is really, really, really bad in your DNA. It messes a lot of stuff up. Yeah. So that's going to be something that in processed RNA, you're going to see a lot of methylation sites. DNA, hopefully not, because that means practically that you have cancer. So that's bad. Bad. Okay. let's see. And so this is the same. Hindi 3, you're going to have one cut site. PS2 1, you're going to have one cut site. You're going to subtract the 2 to get the bands. T5 exonucleases. Hindi 3. That's just going to be an exonuclease, so it's going to cut the ends, which isn't going to do anything since it's circular. Removal of the enzyme, which is going to take off the enzyme, then a Hindi 3. So you're going to have exactly 5,000. 7, Hindi 3, so it's going to be cut here. Removal of the enzyme, so it's not going to have anything attached, and 5, T5 exonuclease. So what would you have? So if you cut it once, take everything out of it, and then stick it on an exonuclease. So Hindi here, you're going to cut once, so you're going to have your whole 5,000 strand, right? Then removal of the enzyme, so that's going to prevent. Where is your enzyme? I guess you don't know where the enzyme is. What's that one? So on this one, I actually would say that it's shifted down, because when you remove the enzyme, it's going to remove the shift, right? So if you cut it and then remove the shifting agent and then stick it in the blender, essentially, you're going to get 0 from 5,000. And so I think you should shift it down. But I've never seen that before, so that's what I would have said. Versus just Hindi 3 and T5 exonucleases, you're going to have just a 5,000 length, but it's going to be shifted up because you have your enzyme in there. Remember when we were doing the fingerprinting analysis and the shift? That's what I'm saying here. Whenever you took out the enzyme, you're going to have no shift, versus, because it's only 5,000 total, there's a 5,000. Versus if you have the enzyme, it's going to be shifted up. So, I don't know, the TA graded this one, it looked like, so. I don't know, another good thing to ask him. Tell him that's what I said, and he makes it well. He's so wrong, but it's just something to think about. Okay, so cat box. That's DNA, you just have to memorize it, sorry. Shindogano is RNA, RNA alpha helix is going to be, what would an alpha helix be? Could you have alpha helix and RNA, you think? I don't know. What would you have? I don't know, that's a good question, I don't know. I would say neither. But Shindogano and Catbox are sequences. They're in the textbook. This is going to be neither because they're amino acids. This one, why was it RNA? It's underlined. Yeah, there you go. 5' cap, we just mentioned it, is going to be RNA. Delivered by AAV, that's a adenosine activated virus or something like that. But that's going to be your that's one of your DNA transduction tools in this thing that you didn't talk about. And these are your things you have to memorize. I'm pretty sure this is U. And this is T, so this is going to be DNA. And why is this one RNA? Yeah, because the OH there. Do you remember what number this is? Which carbon it's attached to? Yeah, so this corner is going to be 1. So right here is going to be 1. Yeah, so it's your 2' carbon. Actually. What is this one? Oh, okay.
RNA.
Yeah, why is it RNA? Yeah, what goes here usually? Yeah, yeah. Well, the base is kind of R group, but this is where you're polymerizing. So this is where your O attaches. You have your phosphate group attach. Remember, so you're not. This is going to be your polymerization one. And let's see, the next one, neither. Why is it that? So I would say I would have said neither, unless you have a specific charge. So let's say you have. Let me think. Let's say your pH is really, really, really crazy, and so whenever you have a really, really, let me get this right in my head. pH is when you have a really, really high pH. Yeah, no, low. Whenever you have a low pH, remember in your amino acids, the pKa, they had the charge on them. So if you made it the pH high, then you would take out. Yeah, then you would take out a hydrogen. So this could be what happened here, is that you changed your pH and made that a double bond. So because it has an oxygen, I would say RNA, but my first guess would have been neither also. So good for her. I agree.
That was a trick.
Yeah, but because of the charge, it probably is RNA. For this question, write the correct term, the product of reverse transcription. We haven't talked about that yet. So, chloroquine. Oh, that's cool. Chloroquine, you haven't talked about that, but it's really, really cool.
Is that the malaria?
Yeah, I think it is. It's the one that prevents, it essentially like opens everything up, all the transporters. I think it is the malaria vaccine, but it's a technique to get DNA into the cell because it essentially opens like all the transporters up and you can put stuff in pretty easily. Yeah, it raises the transfection because they're opening it up. And you haven't learned about that yet. So, the rest, lipids, dope, and there's dope and dogma. He didn't talk about that last class. The plasmid protocol, and that's from last class, so you could do that. Complete the palindrome sequences. You're good with that. It's going to be GAA, and you know when GAA. And then ATC, ATC. You know this one has to be T because of A, blank. So one of these this one has to be a C because it's like that. You see how she did that? It's tricky, just like a little puzzle. How is it possible that 4 different codons can code for the same amino acid? What would the 4 codons have in common? So this is, remember, oh, do you have to go or anything? Okay, cool. So do you remember what the wobble?
Do you have to go?
Oh no, that's fine. Do you remember the wobble hypothesis?
Like the thing like that.
Mm-hmm. Yeah, yeah. So
I'm really bad at articulation
thing. Oh, that's fine. So one thing to mention there is the wobble hypothesis. And so what two would be the ones that are going to be the same? What two? Yeah, the first two are usually the same. How is it possible that 4 different codons can code for the same amino acid? And that's just because there's a word for it, but DNA replication or RNA replication isn't like redundant. There are redundancies in the code, and so that's why it codes for several. For the same amino acid, what would the 4 codons have in common? And that's your wobble. So you can talk about wobble, and then redundancy. It's a bolded word in the text. So watch out for that. Redundancy is pretty cool. Let's see. 2 parents of type O blood produced a baby boy in 1966. Whoa. And this boy eventually met and married a lady with type A blood, and they had a child named Chris in 1991. Oh yeah, that's about right. In 2011, Chris got sick and needed a blood transfusion. What blood type could Chris definitely receive and not receive? So you haven't gone over this, but it's just another, what's it called? Punnett square kind of. So two parents where they're type O blood. So parents are both going to be OOOO. So the baby's going to be OOOO. And the boy eventually met and married a lady with type A blood. So the type A blood can either be two big A's or it could be an A or an O, the recessive O. So if you have, so the baby that they had, or the baby that the first people had, is going to be OO because the parents are both OO. Because O, do you know, like, are you familiar with blood? O is like the most recessive, so if you have like AO or BO, then they're going to be A or B. So if you have O, then it's OO for sure. So that's what their kid is an OO, because they're both O. And type A blood, so you can have either AO, or you could have AA. That's AO is going to give you A, right? And so is AA, so you could have AO, AO, O. Or you could have, this is all going to be A. So, from this one, it's going to be AO from that one, or OO. So what blood types could definitely receive and not receive? Definitely he could receive O, because that's remembered like the universal donor. Could not receive anything with B, because he's definitely not anything B. Remember blood type, you could also have AB, but it's always AB, it's not. So if you're B, you're either BB or BO. A, you're AO or AA. So, I guess, yeah. Punnett. Do you familiar with Punnett squares?
Yeah.
Okay, that's all it is. If a gene sequence is an ascending, is more likely to be a cat box or a Tata box. Why? Cat box, just because it is cat box. Sorry. That's just one of the things you have to memorize again that were in the other test. So that's the promoter section. It's good to. It's clearly beneficial to know those numbers. Which have fewer charges or larger DNA fragments, which have more charges and move more quickly through an agarose gel. Why? What would your reasoning be? Go. So, can you see that still? Sorry. You can't see the question either. So, do smaller DNA fragments, which have fewer charges, or larger DNA fragments, which have more charges, move more quickly through the agarose gel?
The smaller ones move faster.
Why?
Because they can get through the thing easier.
Yeah, correct. The charge is only used to pull them. The charge isn't how far they go. So charge is roughly the same for each unit. So consider it, the charge is like a density kind of thing. So your density for charge is going to be the same as one as it is for other. Because say you have a small one with only like a smaller charge, but you have a bigger one with a bigger charge, but the charge density is the same because. Does that make sense? Yeah. Like your units kind of thing? Okay. Smaller DNA fragments? Yeah, you can read it. After transformation, one will select bacteria that will have the desired plasmid by growing them in the presence of antibiotic. However, this is distinctly mentioned in class that antibiotics should not be present just after transformation. Resolve this apparent contradiction, including scientific reading and your answer. What would your answer be? Let me see. Okay. Yeah, like her answer's pretty good. What is your answer? What would you say? So say we have our cells and we put our DNA in them. Do we want to stick them straight into antibiotic? Why not? Okay. So remember, whenever you go through your what am I thinking? What is the word? Whenever you go through replication, so you're gonna have your DNA, your normal plasmid, and it's gonna be going through replication every 30 minutes? Is that what he said in class, or 20 minutes? It's actually 28 minutes. This is what my research is on. 28 minutes, you're going to have another round. So if you put your new DNA that has the antibiotic resistance in, you stick it in the cells and throw it right into antibiotic, it's not going to be the 28-minute time period for the DNA to like start transcribing the resistance. So does that make sense? So it'd be like someone saying, "Oh, here's an antidote to a poison. Now drink the poison first." Like you still have the antidote with you, but if you haven't taken the antidote yet, then you're still going to die from the poison. So our DNA or our bacteria, we have to let them drink the antidote first before we poison them, or they will all die. But that makes sense?
Yeah.
Okay. Kind of a morbid comparison, but
It gets the point across.
okay. I guess to that effect, why do we put the antibiotic resistance in there to begin with?
So you know which ones have the plasmid.
Yeah. So you know which ones are actually expressing it, because you can't tell just by looking. Oh, like these are doing well, and those have the ones I want. No, you can't tell. Let's see. After fire and mellow discovered, you haven't. Oh, that's splicing and dicing. We haven't even talked about that. I like the color though. It's cool. Let's see if we go over that. And this is the same as the other guy's test. You have to identify Tata, do 25, and then do the start, and then stop, and then do 2 stops. So memorize those. Important. Entire structure of adenine. Yeah, something to like you got this. The genome of the lecture. What? Do you get this? This is just kind of mathy stuff.
Yeah.
So 31.7 is adenine, which means another 31.7 thymine. So you add those together and subtract from 100, and then split that in half to get either the G or C content. So that's all good. Okay.
So definitely bring a calculator.
For sure, especially if you can't do mental math like me. Like, give me a derivative any day, but represents the G. Oh, and they both sketch. Which A through E represents the gene? What would you say? A through E. What is the gene? Oh, A's not right? One sec.
I'm gonna say A.
It's C. I guess if you consider, I don't know. I would ask him about this one. I would not consider, because if gene you only consider the product, then I would say it's A. But C, I guess it's just because you include the enhancer. But even then, like, you have all these introns. Because a gene, I don't know. Do you remember what the definition of the gene was? Okay, we'll look up the definition of the gene in the book and see if it says include introns. Because remember, introns are like these sequences before and after the promoter. Then definitely, because the enhancer is going to help what in your gene. So remember, the enhancer is going to like fold up, and then it's going to bind to activators and to mediators, and then to the promoter, essentially. So technically, the enhancer is part of the gene because it binds all of that. But yeah, so look at his definition of the gene here. I would say it doesn't include the introns, but he might say it does. What does the arrow represent? That's translation start. Oh. Remember translation. So transcription is yeah, I know this is translation. It's one of the the notations. That's going to be where your AUG is. What do the separate white arrows in region A represent? The separate regions in A. The white regions? Maybe exons? That's what the E represents? What is that plus? Oh, exon. I think that's what he's saying. Hmm. No. The darker regions? Tricky. I don't know. I'm not sure about this one either. Sorry. These are like the Gavi questions where you never really know. I would have said the same thing as her. Technically, they're both exons, I guess. Because after start, then it's just going to be the whole gene. I don't know why exons is wrong here. I would have maybe said exons on both, but I don't know. He might have talked about this in her class that we didn't go over. Okay, suppose enhancer contains several copies of an E-box element. Write the code for one such element below. And it's the cat box, the CAT, or it's the CTTG box, the CATIG. That's what I call it. Any base. Okay. Write the code for such element. So that's just another memorization. They're all kind of like in the same section. It's cat box, the CAAT, and the CATG, which